Welcome back, class. This is Silvana O'Coney with Lecture 8, Antimicrobial Chemotherapy. Now, we know that the control of microorganisms is critical for preventing disease and treating disease. But antimicrobial compounds are also incredibly important tools used in research and also in industry. Now, we've looked at the use of chemical and physical agents, and when disinfecting or sterilising an inanimate object, one naturally has to use procedures that don't damage the object itself. The same is true for treating living organisms. The most successful antimicrobial compounds will interfere with vital processes that differ between the microorganism and the host. Thereby, it seriously damages the target microorganism but harms the host as little as possible. So this lecture is going to introduce the principles of antimicrobial chemotherapy, and briefly we'll look at the characteristics of selected antibacterial, antifungal and antiviral compounds. Since Alexander Fleming's discovery of penicillin, natural antibiotics have been found that can damage pathogens in several different ways, as listed here. They may be inhibitors of cell wall synthesis, they might be protein synthesis inhibitors, they may be metabolic antagonists, or they may inhibit the synthesis of nucleic acids. The most selective antibiotics are those that interfere with bacterial cell wall synthesis. Drugs like penicillins, cephalosporins, and so on have a high therapeutic index because they target structures in the microorganism that's not found in eukaryotic cells. Most of the penicillins, which are produced by the Penicillium fungus, as pictured here, are derivatives of an acid called 6-amino penicillinic acid, and they differ from one another with respect to the side chain attached to the amino group. The most crucial feature of the molecule is the beta-lactam ring, which is essential for bioactivity. There are penicillin-resistant bacteria, and they're resistant because they produce an enzyme called penicillinase, also called beta-lactamase. And this enzyme inactivates the antibiotic by hydrolyzing a bond in the beta-lactam ring. Here we have the structures and characteristics of representative penicillins. All of these are derivatives of 6-amino penicillinic acid. In each case, the purple shaded portion of penicillin G is replaced by the side chain indicated. The beta-lactam ring is also shaded in blue, and you can see an arrow pointing to the bond that is hydrolyzed by penicillinase. Penicillins G and V, the first two shown here, are naturally occurring, and both are narrow spectrum. Penicillin G is acid sensitive, so that means it's destroyed by stomach acid and has to be injected. Penicillin V is more acid resistant, so it can be given orally. So the mode of action of the penicillins is thought to be the inhibition of the last step in bacterial cell wall synthesis. And this step is called transpeptidation. So penicillins prevent the synthesis of complete cell walls, and this leads to lysis of the bacterial cell. Penicillins act only on growing bacteria that are synthesizing new peptidoglycan. Here are some others that inhibit cell wall synthesis. The cephalosporins produced by the fungus Cephalosporium are structurally and functionally similar to the penicillins. Vancomycin, produced by Streptomyces, and an interesting one, teicoplanin, which is similar to vancomycin. Teicoplanin was actually first approved for use in Italy in 1987, and it's effective against both gram-positive and gram-negative aerobic and anaerobic bacteria. It interferes with cell wall synthesis, but it does this at a site that's different from that affected by beta-lactam antibiotics. It's a true fermentation product produced by actinoplanes, and it's marketed as Targacid. But because the composition of the product varies depending on the fermentation process used and the efficiency of the process, there's a significant difference between batches of the drug in terms of both concentration and shelf life. So as recently as 2013, processes were still being optimised for the production and reporting on teicoplanin. Now let's look at the protein synthesis inhibitors. Many of the antibiotics inhibit protein synthesis by binding with the prokaryotic ribosome. And because these drugs discriminate between prokaryotic and eukaryotic ribosomes, their therapeutic index is fairly high, but not as high as that of the cell wall inhibitors. Some of these drugs bind to the 30S or small ribosomal subunit, while others attached to the 50S or large subunit. Several different steps in protein synthesis can be affected. So for example, transfer RNA binding can be inhibited, peptide bond formation can be affected, messenger RNA reading, and translocation. The aminoglycoside antibiotics all contain a cyclohexane ring and amino sugars. Examples such as streptomycin, kanamycin, neomycin, and tobramycin are synthesized by different species of the genus Streptomyces, whereas gentamicin comes from another actinomycete called Micromonospora purpurea. Aminoglycosides work by binding to the 30S ribosomal subunit and interfere with protein synthesis by directly inhibiting the synthesis process and also by causing misreading of the messenger RNA. These antibiotics are bactericidal and tend to be most effective against gram-negative pathogens. However, aminoglycosides can be quite toxic, causing deafness, renal damage, loss of balance, nausea, and allergic responses. The tetracyclines are a family of antibiotics with a common 4-ring structure, as shown here, and a variety of side chains are attached to the structure. Some of these are produced naturally by Streptomyces species, while others are semi-synthetic drugs. These antibiotics are similar to the aminoglycosides, and they work by combining with the 30S subunit of the ribosome. This inhibits the binding of the transfer RNA molecules to the A site of the ribosome. Here are some more examples. The macrolides, which contain 12 to 22 carbon rings linked to one or more sugars. For example, erythromycin. Another example is chloramphenicol, which was originally produced from cultures of Streptomyces venezuelae, but it's now synthesized chemically. Like erythromycin, this particular antibiotic binds to the ribosomal subunit. It has quite a broad spectrum of activity, but unfortunately is very, very toxic. So it's only used in life-threatening situations. Let's move on now to our next group of chemotherapeutic agents. And this group belongs to the class known as metabolic antagonists. That is, they antagonize or block the functioning of metabolic pathways. They do this by competitively inhibiting the use of metabolites by key enzymes. So these are drugs that act as structural analogues, molecules that are structurally similar to naturally occurring metabolic compounds. These analogues compete with intermediates in metabolic processes because of their similarities, but they're just different enough so that they prevent normal cellular metabolism. Let's look at an example. The first metabolic antagonist to be used successfully as chemotherapeutic agents were the sulfonamides, or sulfur drugs. And the sulfonamides are structurally related to sulfanilamide, which is an analogue of a substance called p-aminobenzoic acid, or simply PABA, P-A-B-A. Now, PABA is used in the synthesis of the cofactor called folic acid. And when sulfanilamide or another sulfonamide enters a bacterial cell, it competes with PABA for the active site of an enzyme which is crucial in the synthesis of folic acid. And this results in a decline in folic acid concentration. This decline is detrimental to the bacterium because folic acid is a critical precursor of purines and pyrimidines. These are the bases used in constructing DNA, RNA, and other important cell constituents. So the resulting inhibition of purine and pyrimidine synthesis leads to a cessation of protein synthesis and DNA replication, and the pathogen dies. Now, sulfonamides are selectively toxic for many pathogens because these bacteria manufacture their own folic acid. They can't effectively take up this cofactor. Whereas humans don't synthesize folic acid, we have to obtain it in our diet. So sulfonamides have an effect on the bacteria, but not on the humans. So sulfonamides have a high therapeutic index. Here we can see the chemical structure of sulfanilamide, PABA, or p-aminobenzoic acid, and folic acid. So remember, the sulfonamides are structurally related to sulfanilamide. That is, they're structurally very similar to sulfanilamide. Sulfanilamide is an analogue of p-aminobenzoic acid, otherwise known as PABA. And PABA is used in the synthesis of the cofactor folic acid, which is also pictured here. Our next group of chemotherapeutic agents work by inhibiting nucleic acid synthesis. So they actually function by inhibiting DNA polymerase, DNA helicase, or RNA polymerase. So this effectively blocks processes of replication or transcription respectively. These drugs are not as selectively toxic as other antibiotics because prokaryotes and eukaryotes don't actually differ a great deal with respect to nucleic acid synthesis. Here we have an example, the quinolones. These are important synthetic antimicrobial agents that inhibit nucleic acid synthesis. They're increasingly used to treat a wide variety of infections. The first quinolone, called nalidixic acid, was synthesized in 1962. And since that time, generations of fluoroquinolones have been produced. 3 of these are currently used in the United States, and more of the fluoroquinolones are being synthesized and tested. Ciprofloxacin, otherwise known as Cipro, gained notoriety during the 2001 bioterror attacks in the US as one treatment for anthrax.